Hi everybody, my name is Brittany Goodwin, and today we're going to be demonstrating how to do a body tissue culture. So what is a culture? A culture is taking a specimen and taking a sample of it and putting it into an artificial environment to see if any bacteria grows. So today for our artificial environment, we have culture plates. These culture plates all are made with sheep's blood, and they have a little jelly agar at the bottom, and this is what grows the bacteria. They each have different chemicals in them to grow certain bacteria or stop the growth of other bacteria, and this is what we're going to use and then incubate to see if you have any infections. So the first step in doing this is to gather all your supplies. Obviously, since I'm simulating this in my home environment instead of a lab, I have all my supplies ready here. And for our specimen today, we're going to be using a strawberry instead of a real one. Now, if I were using a real specimen, I would obviously have a lab coat on, I would have a mask, and I would be doing this under a biohood so that I don't get any infections myself. The next step is going to be to grind your tissue specimen. So this is a clear, sterile plate. We're going to open it and we place our specimen inside. And since this specimen is far too big for what we need it for, we're going to use a disposable scalpel. And yes, this is a real scalpel, and I will be disposing of it properly into a sharps container afterwards. And we're going to cut different sections of this specimen off so that we can have a wide variety of the specimen inside of our cultures. So for example, I'm going to take some of this dark red area off the top. I'm going to take some of this light pink white area off this side. And I'll take some of the leaves off as well, just for a little bit of diversity. And we're looking for specimens probably no bigger than about that size. And now that we're done, we can put our scalpel to the side and our specimen as well. And now from here, we get to use the best part. So here I have my tissue grinder. I have already put some of this broth inside of this. This is thio broth. It has lots of nutrients that bacteria love, so that it's encouraged to grow, and we use this along with the specimen to grind it up and basically get kind of like a specimen juice. So what I'm going to do is take the top off of this, and we're going to use a swab to gather our specimen and put it inside of our broth. And now we're going to place this in here and screw on the top. So on the inside of this pointed part, there are little grooves that help grind the specimen, and attached to that is almost a kind of pumice stone, and this is what actually grinds the specimen. So now that that's all nice and tight, I'm going to lower the plunger into the liquid slowly so that it doesn't splash me, because that would definitely not be a fun day. And I'm going to use this handle to manually grind the specimen. So this can take a lot longer depending on what kind of specimen you have. Usually what we get is soft tissue, but we do also get bone tissue, which is a lot harder to do. So now that that's done, I can lift the plunger and unscrew the top. And I'm going to place this to the side. And now what we have left is our specimen juice. So I'm going to take a pipette and I'm going to suck up the juice that's in here. And what we're going to do is open all of our culture plates. And these are in order from the least amount of chemicals to the most amount of chemicals, because we don't want to contaminate any of them. We're going to put a couple of drops onto each plate. And now I'm going to cut to a different view so that you can see how I spread the specimen all over the plates to get the best sample growth. All right, so from this angle with my video helper, we're going to plate the specimen. So as you can see, the specimen is all right here where I put the droplets. And now we're going to take this metal loop and we're going to what's called streak the specimen. So I'm going to take my metal loop and I'm going to drag it through the specimen back and forth. And we do a method for these called 4-quadrant streaking. So now that I've spread out my specimen, I'm going to rotate my plate, hopefully so that you can see it. And now I'm going to take my loop and I'm going to go in and out of that streaking that I just did. And then all the way to the other side. Now I'm going to turn my plate again. And I'm going to go into the streaks that I just made. And I'm going to streak again. And now I'm going to turn it a final time. And I'm going to go into what I just did once. And then make streaks down the rest of the plate. And so you can see the specimens over here in the light. Here's my 2nd quadrant of streaking. Here's my 3rd quadrant. And this is my 4th one. And this 4th quadrant will be the one that has the most isolated colonies. So it'll have little specks of bacteria that are easily identifiable so we can tell you what kind of bacterial growth you have. Now for the final step, we also place a drop of our specimen onto this glass slide, and we're going to use this to look at the bacteria underneath the microscope. So what we're going to do is take that drop and use our pipette to slide it all over the plate. Get it spread out nice and evenly and nice and thin, and then let it dry. Now our last step is to take the little bit of specimen that we have left, and we're going to use the pipette and drop it inside of this broth. And we're going to seal it shut. We're going to shake it up a little bit. And then we're going to put it in the incubator with the cap loose, because when bacteria grows, it creates gases. And when you open that up, and if there is gases in there, it will not be very pretty. So that is how you plate a body tissue culture. You're going to take all of your plates, stack them up nice and neatly, and put them all in the incubator.